कुछ निकला सॉल्यूशन?
हाँ सर सर, मेरी बात होगी सर से। सर बोल रहे हैं कि मैं ज्वाइन कर रहा हूँ लेकिन कनेक्शन एरर आ रहा है।
हम्म।
अब मैं ये नहीं पता सर क्या इशू आ रहा है। मैं या तो उनको सर लिंक फिर से सेंड करता हूँ, मैं शेयर करता हूँ लिंक।
हम्म। रोहित जी, आप भेजो जरा यार, वो क्या भेज रहे हो आप, वो भेजूं फिर मैं उनको?
उनको लिंक भेज दिया सर मैंने ऑलरेडी।
उनको फोन भी करके कह दो।
जी सर।
फोन तो करो महाराज, बिना फोन करे कैसे वो जानेंगे? कोई सफलता मिली?
हाँ सर, बात हुई मेरे सर से। कॉल किया था मैंने, उनको बोला कि आप फोन से ही ज्वाइन कर लीजिए।
हाँ हाँ, फोन से वो कर लेते हैं, फोन से काफी कंफर्टेबल रहते हैं। पीपीटी आप शेयर कर देना, ठीक है?
जी सर, जी सर, जी सर।
हम्म।
अंकुर सर, मेरी स्लाइड्स दिख रही हैं?
रोहित जी वो कर ही नहीं पा रहे हैं, क्या करें इसको, गूगल मीट से करें क्या?
सर मैंने सर को ये बोला कि दूसरे डिवाइस से ज्वाइन कर लीजिए। उनका कनेक्शन
हम्म।
एरर आ रहा है, या तो सर नेटवर्क इशू होगा लग रहा है। क्योंकि
यार
मैंने
फोन
सेम लिंक एक
कर,
फ्रेंड को
उनके
भी।
साथ हमेशा ऐसा ही रहता है, कमिट कर देते हैं फिर ये काम करते हैं, सारे फैकल्टी पार्टिसिपेंट ज्वाइन कर चुके हैं, उनके साथ ऐसा हो रहा है।
मैंने
एक
सर वो
बार आप
क्रॉस
उनको
चेक।
गाइड करिए, नहीं तो फिर क्या किया जाए, उनसे बात करिए प्लीज आप।
सर मैं 2 बार कॉल कर लिया उनको, बात भी करी, लेकिन सेम इशू आ रहा है। अभी सर को
अच्छा।
बोला कि दूसरे डिवाइस से ज्वाइन करने के लिए भी देखते हैं सर क्या होता है।
प्रगति जी को बना दीजिए को होस्ट। मैंने बोला है किसी दूसरी डिवाइस से ज्वाइन करिए, अपनी वाइफ के फोन से ज्वाइन कर लीजिए, मैंने सबसे बोला है।
हाँ हाँ सर, बताया सर ने।
हाँ। एटलीस्ट कमिट करने के बाद ये सारी चीजें फैकल्टीज को देखना चाहिए।
यस सर।
नंदिनी एक बार आप कॉल करो जरा।
यस सर।
नंदिनी।
ओके सर, डूइंग।
बात हुई?
सर मैं उनके साथ फोन पर ही हूँ। एक्चुअली ही इज हैविंग सम काइंड ऑफ प्रॉब्लम।
अभी किसी ने ज्वाइन किया था जो बोल रहा था, अभी किसी के नाम से हुआ था। आपने कुछ गाइड नहीं किया उनको?
नहीं नहीं सर, वो कॉल पर ही मैंने उनको बोला कि कोई आसपास अगर कोई है तो आप उनसे हेल्प ले लीजिए। वो फोन 2-3 फोनों से नहीं कर पा रहे हैं वो।
हम्म।
सर एक्चुअली उनको समझ नहीं आ रहा है।
नहीं कर पा रहे हैं ये।
Meanwhile, Sar is joining. So I wanted to give an important announcement to everyone. Before we begin this final lecture, Sar is joining. There is some kind of network issue or something like that. So I'm very happy to share an important announcement from AgriMeet Foundation Bharat as we successfully conclude the ICR Winter School Bharat 2025. We are moving ahead with another enriching learning opportunity, a 10-day agri workshop come certificate course, Mati 2025. This workshop is highly cost friendly and designed especially for students, research scholars, young professionals, and anyone who wants to upgrade their skills in agriculture. It will focus on modern approaches in agriculture for
Faculty
transform.
and scientists also for their case and
Yes,
for their career advancement scheme and direct recruitment.
yeah, it will focus on modern approaches in agriculture for transforming Indian soils, covering practical and emerging domains through expert led sessions, notes, videos, ICR, NEP based curriculums, certificates that support API, CAS, and direct recruitment. The workshop will be starting from 5th, that is World Soil Day, to 14th of December 2025. You all are requested to kindly join, and the last date for registration is 5th of the December. Thank you so much.
Pandyan sir, I'm audible to you. Pandyan sir, please unmute yourself. You can unmute yourself, please. Sir, I'm audible to you now.
आपको दिख रहे हैं? वो हमें तो नहीं दिख रहा है। किस नाम से ज्वाइन किए हैं?
सर ज्वाइन किया उन्होंने, लेकिन नाम का नहीं पता, लेकिन नाम ज्वाइन कर लिया सर ने।
अच्छा,
मैं उनसे कॉल करके पूछूं क्या?
हाँ मैम, प्लीज जस्ट।
हम्म, हम्म।
Pandyan sir, I'm audible to you. Please say something. Say hello or something.
नंदिनी क्या बोला उन्होंने?
सर उन्होंने ज्वाइन कर दिया है, कोई सेंथा मिल नाम से ज्वाइन किया है।
मैम किस नाम से ज्वाइन किया सर ने?
सेंथा मिल बोल रहे हैं, वो इस तरीके से बोल रहे हैं।
हाँ
उनकी
मैम,
वाइफ
लेकिन
भी
आ नहीं
बोल,
रहा। मैंने सर्च किया और आ
हाँ,
नहीं रहा।
आ नहीं रहा है, मैंने भी सर्च किया है।
Pandyan sir,
वेट,
you can unmute yourself. Please click on that mic icon and
वो सुन
type.
पा रहे हैं हमें।
वो सुन पा रहे हैं हमें?
Yes sir, join
हाँ
कर
जी,
लो।
हाँ जी, हाँ जी।
OnePlus 12 को होस्ट बनाया आपने, ये कौन है?
सर है सर ये।
अच्छा, इनको अनम्यूट कर दो आप ही। यार कितना वेट करना पड़ा, इतना हमने किसी वाइस चांसलर का वेट नहीं किया।
हेलो।
हेलो, हाँ डॉक्टर साहब, उम्मीद की किरण
हाँ,
जग गई है
निस्तम
अब।
सिस्टम्स।
चलिए, आप
हेलो।
आए तो बाहर आई। आवाज आ रही है आपकी, आपको?
ओके, कैन आई प्रोसीड?
यस सर, यस सर।
अब नहीं करेंगे तो कब करेंगे महाराज?
सर चालू कीजिए सर।
नो नो, दिस इज अनदर फोन। ओके
एक
ओके।
फोन को म्यूट कर दी, एक फोन को म्यूट कर दीजिए, नहीं ऐसे ही बोलता रहेगा वो, दूसरे वाले को म्यूट कर दीजिए।
डन सर, डन डन।
हाँ
हेलो।
जी
हेलो
जी,
सर।
प्लीज कंटिन्यू करिए आप। रोहित जी पीपीटी
सर
दे
मैं
दीजिए
स्टार्ट
आप।
करूं?
बट पीपीटी इज नॉट डिस्प्लेड मी।
नंदिनी रहने दीजिए, स्टार्ट करने दीजिए सर को। सर प्लीज स्टार्ट करिए आप।
हाँ, यस
ओके
सर।
ओके ओके। आई एम डॉक्टर पांडियन, प्रोफेसर, plant breeding and genetics, former dean, Dr. M.S. Swaminathan Agriculture College and Research Institute, TNAU, Ichamote, Tanjavur, Tamil Nadu. My topic for this session is breeding for drought tolerance mechanism in crop plants. So stress, drought means stress. Stress is unfavorable condition that affect or blocks plant metabolism, growth, development. So in general, it comes under abiotic stress. It is abiotic stress is negative impact on living factors on living organism in a specific environment. So characteristics of abiotic stresses, it is unpredictable occurrence. Sometimes some stresses are impossible to manage. It is differential response of plant species to a given stress, and the effects generated by one abiotic stress may overlap with some other effect of other stress. Generally, two types of stresses, biotic stress and abiotic stresses. What topics I am going to tell is comes under abiotic stress. Biotic stress reference to pest and disease. That abiotic stress, they are some metal toxicity, high temperature, light chilling, cold, drought, salinity, submergence. These all comes under abiotic, major abiotic stresses. Now I am going to deal drought tolerance. So losses caused by the abiotic stresses, the progressive salinizations of soil. The salinizations also comes under a drought condition, estimated at around 20% of irrigated land in world. It was estimated that approximately 70% of yield reduction was due to abiotic stress. The estimated potential losses in general, 17% due to drought, 20% due to salinity, and 40% due to high temperature, 50% due to low temperature, 8% due to other stresses. So major challenges towards world agriculture involves productions of 70%, more food crops for additional 2.3 billion people by 2050 worldwide. Abiotic stresses governed by mineral salts affect considerable proportions of the arable land, and they rank second after moisture stress. So drought stress tolerance and mechanism in crop plants. Now I am going to deal two portions. One is general for drought mechanism, and another one is what I have done in my research station during 2017-16. In that period, I have done some of the work related to drought mechanism and uh, uh, drought tolerance. So what is drought? Drought is a period or conditions of unusual dry weather with a geographic area where there is a lack of precipitation. Drought tests account for more production losses than all other factors combined. Fraction of world arable land subjected to drought abiotic stress, 26%. Drought is governed by various factors. The most prominent are extreme of temperature, potent irradiates, paucity of water. Then high drought affects plants. They are water stress, iron toxicity, nutritional disorders, oxidative stress, alterations of metabolic process, membrane disorganization, reduction of cell division and expansions. World scenarios related to drought. It's affect 35% of the world agriculture land. Drought prone approximately 26% of world arable land. Its limit more than 50% of the world agriculture productivity. Due to drought and desertification, each year 12 million hectares are lost, where 20 million tons of grains could have been grown. Materials, methods, so related to drought. So material is concerned, different crop species. Method is concerned, stress defense mechanism by plant. Second one is drought stress mechanism in plant. Third one is drought effect on plants. These information is collected from the secondary sources, journals, books, theses, online also I collected. The first one is plant defense mechanism by plant. So plant stress defense mechanism, plant itself having the capable to control, to able to produce productive and yielded in the drought conditions. Even though ROS are present in the plant system through enzymatic and non-enzymatic actions, that is defense mechanism, plant itself having the capable to control the ROS. ROS, you know, one is the 1 oxygen and 2 oxygen, then hydroxide and peroxides. These are all the ROS species which damage the plant system under drought conditions. Because of enzymatic SOD, CAT and GPX, GR, non-enzymatic ascorbic acid, GSH, glutathione, GSCS and DHA. These are in the balanced condition, the normal condition. Normal conditions, the plants are growing well, and under the plants going under drought condition, these are all the enzymes help to the plant system to arrest the or control the ROS. So that is plant stress defense mechanism. Then drought stress mechanism in plant. Drought stress mechanism in plants. Drought stress, when water availability is reduced, ABS signaling to the plant system, and stomatal close, immediately stomatal closure is happening. It's followed by diminishing CO2 influx. CO2 influx into the plant system is diminishing, then ROS production is more, then oxidative stressors coming. Then third one is drought effect on plant, ROS effect on plant. In ROS effect in the plant system during the drought condition, what will happen in the plant? But the first one is protein degradation. Second one is enzyme inactivations, DNA damage, lipid peroxidations. So DNA damage happen in that, automatically all biological systems are hampered. Then the characteristic features of drought stress is the first one is the low water potential due to high solid concentration. So low water supply cause soil mineral toxicities and can make a plant more susceptible to damage from high irradiance. The major reason for drought, atmospheric factors, is related to high temperature, high wind, air pollution. Soil factor is concerned, low temperature, excessive soil salinity, and residing water content. So different types of drought. So meteorological drought, 25% of average rainfall below that area is due to prolonged period with less than average precipitations. If there is no rainfall or less than 20% of the rainfall received in the area, this comes under meteorological drought. Agricultural drought, which affect the crop productions. That means surface soil and subsurface soil moisture depletion. This is prolonged due to prolonged rainfall, non-availability of rainfall. So hydrological drought, when the water reservoirs available in the sources such as lake and reservoirs fall low statistical average. They are water reservoir, so lake also depleting water, river also getting depleting waters due to continued drought conditions. This is hydrological drought. So effect drought stress on plants. Effect on growth, reductions of turgor pressure, cell size will be also smaller. So because of this water problem, so effect on photosynthesis, photosynthesis decrease due to disruptions of photosystem 2, stomatical closures and decrease in electron transport. Decrease in nucleic acid and proteins, the protease activities, RNase activity, RNA hydrolysis, and DNA content falls down. The effect on nitrogen metabolism, on nitrate reductase activities, insensitive. Then the effect on carbohydrate metabolism, loss of starch, carbohydrate translocation decreases. These are all the things happen due to drought stress on plant. Then mechanism of drought tolerance. The first one is drought escape. Tolerance, that previous one is resistance. The drought escape, it is defined as ability of plant to complete its life cycle before supply of water in soil is depleted. This plant known as drought escapers. Drought avoidance, it is ability of plant to maintain relatively water balance despite and shortage of soil moisture. Then drought tolerance, it is ability of plants to withstand water deficit with low tissue water potential. This all comes under drought tolerance mechanism. So mechanism of resistance to drought. Yeah, the first one is morphological response to drought. Increase in water absorption and transportation, declarations of transpirations. That is related to developed root system and high ratio of roots to shoot, thick leaves, smaller, the plants having smaller leaf area, and thick cuticle, developed veins and bundles, smaller and more stomata. Then root system development, the plant grow larger and deeper root system to increase water uptake from the soil. Leaf adaptations, also one of the main related to drought tolerance. Leaves may become smaller, thicker, develop feature like waxy cuticles, shrunken stomata, hairs, that is pubescence to reduce water loss. So water storage, some plants can store water in specific organs to endure prolonged drought conditions. Leaf shedding, certain plants shed their leaves during severe dry period to minimize transpiration. So drought condition, so ethylene is doing the same characters for some of the leaves shedding. Whenever drought comes, plant automatically to shed the leaves. Then physiological mechanism. So physiological mechanism, so it is comes under osmotic adjustment. So plant accumulate compatible solids such as proline, sugars, and other organic acids in their cells or lower the cell's water potentials, helping to maintain turgor and prevent cell collapse. So antioxidant defense. Antioxidants defense. The plants activate what are the antioxidants? I'll tell you later. The plants activate their antioxidant system to scavenge the detoxifying reactive of the cell species, which are harmful byproduct of stress that can damage cells. So stomatal regulation. Hormones like ABA produced in response to stress trigger closure of stomata, thereby reducing water loss through transpirations. So you know hormones. There are 5 major hormones. One is cytokinin, auxin, gibberellin, abscisic acid, and ethylene. The abscisic acid, ethylene, the two hormones out of 5 hormones, abscisic acid and ethylene comes under stress hormones. So ABA, abscisic acid, always helps to plant for withstanding the drought situation to produce more number of roots under drought condition, under severe drought conditions. Best example, sorghum plants. During a drought condition, sorghum produced more number of roots, more number of aerial roots. Then molecular mechanism, hormonal signaling, phytohormones, including abscisic acids, are key players in signaling drought stress and activating the plant defense response. Synthesis of protective proteins. The plants synthesis protective proteins such as dehydrins, Lea proteins, and heat shock proteins to stabilize cellular component and maintain membrane integrity under stress conditions. Then gene regulation. The transcription factors behave to specific DNA sequence to regulate expressions of genes involved in drought tolerance, leading to productions of necessary protein and metabolites. Another things, aquaporine regulations. The plant can modulate the activity of aquaporins. Aquaporins mean water channel proteins, which are crucial for water transport and maintenance of cell-water balance. Then physiological and biochemical basis of drought stress tolerance in plants, increasing capacity of resistance to dehydration of cytoplasm, rapid accumulations of proline, then glycine, betaine, and other ions. So in this, synthesis of specific protein like Lea protein, reduced ROS accumulation, increase in antioxidant enzymes, decreased efficiency of Rubisco. So phytohormones plays vital role in drought tolerance in plants under drought conditions. Endogenous content of auxin, gibberellin, cytokinin usually decrease, while those of abscisic acid and ethylene increases. So auxin, gibberellin, cytokinins, this is a normal condition, it is increases. And unusual conditions, especially drought condition, it is decreases, while abscisic acid ethylene increases to help the plant system for withstand under drought conditions. There are many signals that induce stomatal closure. Among these best known signal is abscisic acid. So drought tolerance in crop improvement. There is a high correlation between tolerance to heat and tolerance to desiccation. Comparatively, resistance of plant genotypes may be observed by exposure to high temperature, soil drought, or atmospheric drought. Like winter hardiness, plant genotypes resistant to heat and drought stress in the field is determined by the complex physiological and morphological characteristics and cannot be accurately evaluated by the single laboratory test. Heat and drought stress resistance are quantitative characters with complex inheritance breeding for drought tolerance. Then conventional method based on genetic variation and selection. Second one is molecular breeding, biotechnological techniques based on tag and molecular markers. To develop drought tolerant genotypes, appropriate selection criteria must be decided on. The extreme expressions of phenotypes of traits of interest is not always desirable and cannot be used as a best selection criterion in crop improvement program. So up to this, it's a general things I'm telling. I told you already, two part I'm going to explain. The one is general things, another one is what I have done in the research station. So this is the study I have carried out in my official program. The studies on performance of drought tolerant genotypes under drought and normal condition through morpho, physio, and biochemical attributes of black gram and green gram. So introduction, you not read again. Usually drought condition, the extreme drought condition. So productivity is decreasing. Crop production is completely affected by periodical, regular, continuous drought. Agriculture is the biggest consumer of water. So 70 to 80% water is consumed by agriculture industry. What is the main source involving for all activities of plant growth throughout the life of the plants? Seed to seed production is complete agriculture. Without soil, seed will germinate, but without water, seed will not germinate. That is the main thing. What is essential medium for seed germination? Seedling growth, vegetative period of the growth, flowering, translocate, assimilate throughout the body of the plant, from roots to top of the plant and leaf to bottom of the plants. So aim and objectives: to study the green gram, black gram varieties under water stress and normal condition, to test verify the reality of tolerance and confirmation through morphological, physiological and biochemical parameters. To observe the changes in these parameters during drought condition, to study the overall effect of drought on yield component. Those two set of experiment I have done. The one is completely, so one certain period only we are giving irrigation. After that, there is no irrigation. So one set completely not irrigations. This is the two study I have handled. So materials and methods, 6 genotypes taken for this study. 4 test varieties are again test verify the real drought tolerance under irrigated and drought condition. The VRMJG1 and VMJG 12005 in green gram and 114 and 115 in the black gram. Check variety CO8 in green gram and CO6 in black gram. This trial was laid out during to March 2016 at TNA Agriculture Session, Virinjipuram, Ellore. Irrigations were given as per the schedule, 1st, 7th, 25th and 40th days. No irrigation was given in drought imposed plot for both green gram and black gram. The biometrical characters, namely plant height, number of branches per plant, length of branches and number of pods per plant, pod length, number of seeds per pod, 100 seed weight, seed yield per plant, dry matter productions were also recorded. 10 plants from each genotypes were taken for work out the mean. So soil analysis, soil pH EC taken. Soil moisture was recorded immediately after second day of irrigation at middle of the plot in each in the irrigated plot. Soil moisture content was also determined by the gravity methods at regular interval. Plant morphophysiological analysis, leaf membrane stability also studied. Membrane stability index was determined according to the method of Bremchandra and co-workers, modified by Sairam 1997. So leaf area index, leaves from 10 plants in each genotypes were cut and leaf area was measured in centimeter by green leaf area meter. So plant biochemical analysis, chlorophyll A, B and total chlorophylls were determined. So chlorophyll stability was also calculated periodically using following formula. So proline also, determinations of free proline was estimated using method of Bates and co-worker. The leaf proline content was analyzed at 28 days and 40th days. The obtained data were analyzed using the statistical package PASOW statistics. So result, weather data, irrigation schedule for green gram, black gram genotypes. In the present study, which includes 6 genotypes for experiment and 3 genotypes from black gram and green gram. During the crop period, temperature and rainfall were recorded. There is no rainfall received during the entire period of crop growth. Soil moisture content was analyzed at 4 stages of varietal plot. Every time sample were taken for analysis at second day after irrigation. It is given in the table 1. The both conditions of irrigated, table 2 and drought, table 3. So this is the weather data. So January 15, 2016 and 31/3/2000 during the crop period. So these are the temperatures recorded. The irrigation schedule for green gram and black gram. The first irrigation, second irrigation, third irrigation, fourth irrigation. All the varieties green gram, black gram, 1, 7, 25th and 40. All the varieties green gram, black gram, 1, 7, 0 and 0. After that, there is no irrigation at all. So soil moisture content under irrigated condition. These are all the conditions. To second day, 8th day, 26th day, 41. So these are all the varieties. Soil moisture conditions also we have recorded. A drought imposed condition, soil moisture also is recorded. Effect of drought stress on plant growth parameters. In the present study, plant growth parameters in terms of plant height and all biometric characters, including leaf area, were observed and are also presented in table 4 and 5 and 6. It is comes under next slides. Drought stress has been found to decline the almost all the morphological characteristics in both crops compared to those of irrigated ones. The plant height is reduced, very much reduced. Most of the plant not having branches. Even 1 or 2 plants having branches with shorter and unproductive. The number of pods per plant, pod length, and number of seeds per pod also much reduced. The 100 seed weight, single plant yield, and dry matter productions were also reduced compared to irrigated condition. Susceptible drastically, it is reduced almost all the characters under the drought condition, unirrigated condition. The susceptible genotypes of CO8 in green gram, CO6 in black gram were much reduced in all biometrical characters, including leaf area, compared to water stress tolerant genotypes, which shows susceptible to water stress. This reduction in plant growth in terms of all quantitative characters are due to loss of cell turgor, which greatly suppress cell expansion and cell growth, thereby inhibiting linear growth of shoot observed more in drought susceptible genotypes. Stress tolerant genotypes somewhat better than the susceptible genotypes towards yield. So this is the morphological characters of green gram, black gram under normal condition. So yield is 1.5 gram to 5 gram per plant. Actually, it is the normal condition. So under drought condition, there is no yield is concerned very low in the green gram and black gram. Then leaf area, drought tolerant varieties are having capable of osmotic adjustment to cell alive to protect our longevity and maintain the cell turgor. In susceptible genotype shows reduction in cell division, resulting in reduced cell number and stop cell elongation, inhibiting leaf expansion. Even reduction in complete drought imposed situation and tolerance genotypes have considerable yielding capacity been observed in the all 4 varieties, when same situation imposed in drought and susceptible genotypes completely lower in all the characters. The reductions of leaf area was more prominent in all the varieties that faced the longest period of water deficit, 35 to 55 days. In general, green gram, black gram, and all the pulses comes under somewhat drought condition. Comparatively, cowpea having the more tolerant to drought situations. The effect of drought stress on plant leaf area of green gram and black gram genotypes. So second day, say 8th day, 26th day, 41 day. So drought conditions, the same day we all also are recorded. These are the fact for leaf area in different varieties. So effect of drought stress on cell membrane stability. Considerable decrease in membrane stability in the plant grown under drought stress condition as compared to irrigated plant for both crops. This comes in the table 7. The plant kept under more than 35 to 55 days of water deficit had the lowest membrane stability value compared to plant grown under irrigated condition. So among the genotypes, CO8 in green gram and CO6 in black gram had very low membrane stability. The decrease in cell membrane stability was found to be more in green gram comparatively black gram. So green gram is more stable. The membrane stability index was found to be positively correlated with soil moisture content for both black gram and green gram cultivars. The lowest membrane stability index of CO8 green gram and CO6 black gram accompanied with greatest seed yield loss of this genotypes, while other had highest value of MSI with the normal seed yield. They indicate the underlying physiological mechanism that contribute in water deficit tolerant. So effect of drought stress on biochemical parameters. In the present study, chlorophyll A and chlorophyll content of both black gram green gram plants showed decreasing trends with the increasing durations of drought, which proved that these photosynthetic pigments are sensitive to water deficiency condition. A continuous decrease in chlorophyll stability index was observed during the drought period, more in Co8 green gram and Co6 black gram. So reduction in photosynthesis under water deficit stress can also be attributed to decrease in chlorophyll content. Water deficit reduced the chlorophyll content in water susceptible genotypes and increase in water deficiency tolerant genotypes. So chlorophyll stability index is indicator of the stress tolerant capacity of the plant. So those varieties having stability index more means it is a drought tolerant genotypes. A higher CS helps plants to withstand stress through better availability of chlorophyll by maintaining the more dry matter production and high productivity. The drop down CSI in green gram was faster than the black gram. So membrane stability, chlorophyll A, chlorophyll B, chlorophyll AB, and the carotenoid total chlorophyll, carotenoids in the black gram, green gram genotypes under normal stress conditions. These are all the facts for all the varieties. So proline accumulations help in maintaining the better osmotic balance in plant cells suffering from water deficit. In the present study, more accumulations of proline was reported in leaf tissue with the increase in durations of drought stress. So it is mentioned in the table 8. So regular moisture supply through irrigation decreased the free proline content, whereas reverse proline content is increased in leaf water deficit condition. Varieties having different degrees of drought resistance differ in the capacity of accumulate proline under stress. Recent varieties like VRMGG1 and VMGG012, 005 in green gram and 114 and 116 in black gram had accumulated higher level of proline under water stress. So this is the proline content in different genotypes of green gram, black gram, irrigated and under irrigated and drought conditions. So correlation among characters. And the correlation between the leaf proline content and soil moisture content was found to be negatively correlated. At the same time, correlation between the chlorophyll stability, morphological characters and soil moisture was found to be positively significant in both green gram and black gram. This is the correlation among the characters. So discussion, coming to discussion part. So three component: morphological, physiological, and biochemical. Morphological responses, plant. So most of the studies agree with the already available articles. It's a similar result I have obtained. So three component: morphological, physiological, and biochemical. Morphological response is related to plant drought tolerance, change at the whole plant tissue, physiological, biochemical, and molecular level. Alter the gene expression and molecular level is concerned, alter the gene expression and cellular metabolism to change in growth, productivity, finally susceptible genotypes will die. Major two portions of roots and shoot. Shoot, stem, leaves, petioles, branches, flowers, pods, seeds. Root is concerned, taproot, lateral root, and root caps. All the parts of the plants were affected by the drought in black gram and green gram. So these are seed germination. The first and foremost effect of drought is impaired germination and poor stand establishment. So this is agreed with my result. That is Harris and co-workers 2002. Seedlings. Drought stress has been found in the decline the linear growth of shoot in both cultivars as compared to those untreated ones. This is another authors are agreed with my study. Leaf is concerned, drought tolerant genotypes, leaf area is much reduced. Number of leaves also very less per plant. Size of the leaves are very small and petiole also reduced in length. And some genotypes express clustering of leaves at top of the plant. This is environment factors. So clustering leaves will come during drought conditions. So sometimes actually in a trifoliate leaves in green gram, black gram, some unwanted condition on stress condition, 5 leaflets comes in the one leaf, 5 leaflets. These variations occurred in the some of the genotypes during drought conditions. So less number of hairiness in the leaves. Leaf veins are all not promptly known. So many workers, the same result has given. Stem is concerned, stem elongation completely stopped, reduced in length, reduced internode length, stop phloem growing stems, stop xylem growing stems, reduced axial hydraulic and conductivity was low to negligible. This also found by another authors. Branches also not showing branches. Even branchled types also not produced branching under drought conditions. All genotype shows monostem under drought conditions. So flower also much reduced. Genotypes not flowered. Sometimes tolerant genotypes were flowered at early top of the plant, no pod formation. Generally, sparsely flowering and some of the genotypes get dense flowering. Both are not effectively formations of the pod. Reduction in maize kernel, rose carbs. This is the Yadav and co-workers. This the same things observed in the maize. Pods. Pod is concerned. Even tolerant genotypes are not producing more number of pods. More number of pods. The length of the pods also reduced from original length. Distance in the pod between the two seed cap length of also reduced, changing the original color of the pods, thickness reduced or less hairiness. So the Barua and Gohi, they are also published article, the same pod characters of pulses. Then seed is concerned, seed set in the pods are not normal. In green gram, black grams under normal conditions, the genotypes are having average of 10 and 6 seeds per pod. Some of the genotypes are having 6 seed and 2 per pod under drought situation respectively. Seeds are aborted and genotypes having small size seed pod, but no abortion and seed number is reduced according to the pod length. So this also confirmed with other workers. Root, the same thing. The favorable root system under drought condition. So roots are the only source to acquire water from the soil. So stunted root growth, root caps dried. Root caps dried means there is no, cannot take the water from the soil. Lateral roots grow stopped. Root density also reduced. Proliferations completely stopped. Root system. Architectures also changed. So favorable root system, you know, under drought tolerant genotypes, deep root system, thick root system, and all whatever normal conditions plants acting, the same thing acting in the drought condition only because of drought tolerant genotypes. So dry matter productions also much reduced under drought condition. The yield is concerned, grain yield is reduced, limited size the source and sink tissues. So reduced assimilate translocations, partitioning the activities of sucrose and starch synthesis enzymes. So somatal closure reduced, CO2 to intake result photosynthesis, decline yield. So this also confirmed with other other workers. Favorable function for yield, degrees to which plant vegetative and reproductive growth patterns can be regulated, and function of complex interplay between sources, sinks, limitations of two main organs of the plant, root system and shoot system, establishing functional equilibrium. So this study agreed with many authors. Cell elongation, higher plants can be inhibited by interruptions of water flow from the xylem to surrounding elongating cells. So drought caused impact mitosis, cell elongation and expansion result in the reduced growth, yield Then modifications of leaf anatomy is one of the basic cause which leads to reduction of average leaf size under water limiting situation. Plant height also function of cell divisions and enlargement and differentiation. And red gram is concerned. Totally it's, comparatively red gram distant to drought, even though high severe drought, the plant height also reduced. Plant height also reduced, not flowering, and anything is nothing will come in that. So cytological abnormalities, cell growth is considered one of the most drought-sensitive physiological processes due to reduction in the turgor pressure. So reduced daughter cell productions by meristematic cells, divisions, mitosis, subsequent massive expansions of young cells. Cell elongation, expansion, cell growth also reduced. Nutrient relations under drought condition limited the new total nutrient uptake and the diminishing the tissues concentration in crop plants. So reduced acquisitions of nutrients by the roots and their transport to shoot. Lower absorptions of inorganic nutrient uptake and unloading mechanism. Reduced transpirational flow. A reduced transpiration rate due to water deficit. Reduce the nutrient absorption and emission. So whatever I am doing, the same result explained by the other authors. Physiological mechanism. So already we discussed in the common things. This also somewhat is common. It is useful to drought workers in various crops. So physiological mechanism, osmotic adjustment, osmoprotectant, antioxidants, and scavenging defense system have been most important bases responsible for drought tolerance. Physiologically is concerned, osmotic adjustment, osmoprotectants, and antioxidants, scavenging defense system have been most important bases responsible for drought tolerance. Osmotic adjustment and osmoprotectant. What are the osmoprotectant? The last slide I will given. We will see later. So osmotic adjustment allows the cell decrease osmotic potential, as a consequence increase the gradient for water influx and maintenance of turgors. This is essential for maintaining physiological activities for extended period of drought in maintaining cell and tissue water conservation. With the accumulation of solutes, osmotic potential of cell is lowered with attract water into the cells and help with turgor maintenance. The osmotic adjustment helps to maintain the cell water balance with the active accumulations of solute in the cytoplasm, thereby minimizing the harmful effect of drought. Osmotic adjustment is important traits. Facilitate the better of pre-anthesis carbohydrates portioning during grain filling, while high turgor maintenance leads to higher photosynthetic rate and growth in drought tolerance genotypes. So compatible solutes and osmotic adjustment. Osmotic adjustment is mechanism to maintain the water relation under osmotic stress. Osmosolids. The osmosolids are soluble sugars, sugar alcohols, proline, glycine betaine, organic acid, calcium, potassium, chloride. These are all the minerals, etc. It helps to growth, photosynthesis, and assimilates spotting to grain filling. So one of the most common stress tolerance stages in plant is overproductions of different types of compatible organic solute. Compatible organic solids only give the potential to withstand the particular genotypes under drought conditions. Compatible solids are low molecular weight, highly soluble compounds that are usually non-toxic even at high cytosolic concentrations. So highly soluble and compatible compounds that are usually non-toxic even at high cytosolic concentrations. Generally, they protect plant from stress through different means such as contribution to osmotic adjustment, detoxifications of reactive oxygen species, so stabilization of membranes and native structures of enzymes and proteins. Then photosynthesis hampered gas CO2 exchange by closer stomata because of ROS activities. So photosynthetic carbon assimilation is decreased, change in photosynthetic pigments, decrease in leaf expansion, impact photosynthetic machinery, premature leaf senescence, oxidations of chloroplast lipids and changes structures in pigment and proteins, accumulations of reactive oxygens species which includes induce oxidative stress in protein membranes, lipids and other cellular compounds. Yes. Chlorophyll content. Chlorophyll content, chlorophyll content only gives the greenness of the leaf and photosynthetic, increase the photosynthetic effect. So biochemical mechanism, antioxidant defense mechanism, that is enzymatic and non-enzymatic components. Enzymatic components are superoxide dismutase, catalase, peroxidase, ascorbate, peroxidase, and glutathione reductase. Non-enzymatic components, cysteine, reduced glutathione, and ascorbic acid. So enzymatic antioxidant is a superactive dismutase plays important role and catalyze dismutations of two molecules of superoxide into O2 and H2O2, that is oxygen and peroxidase may directly scavenge ROS. Non-enzymatic oxidant genes such as glutathione reductase, ascorbate peroxidase, and protection of cellular machinery against damage by reactive oxygen species. Non-enzymatic antioxidants cooperation to maintain the integrity of the photosynthetic membrane under oxidative stress. O2 can dismutate into H2O2 by SOD in the chloroplast, mitochondrial cytoplasm and peroxisome. POD will play a key role in scavenging H2O2 which was produced through dismutations of oxygen catalyzed by SOD. CAT also it is eliminate H2O2 in the mitochondria and micro body. Peroximose is also decomposing H2O2 during stress. The capability of antioxidant enzymes to scavenge ROS and reduce the damaging effect may correlate with drought resistance of plants. So reactive oxygen enzymes, it is distract the protein, lipids and DNA, causing the damage and impairing the normal function of cells. This also already explained. Being highly reactive, ROS can seriously damage plants by increasing lipid peroxidations and protein degradation, DNA fragmentation, and also ultimately cell death. Among the enzymatic component are superoxide dismutase, catalase peroxidase, ascorbate peroxidase, glutathione reductase, act on ROS and protecting the cell. So plant growth regulators. Already I told auxin, cytokinin, gibberellin is working well under normal condition. Ethylene and abscisic acid working under well in the drought conditions. The ethylene always doing before before getting the signal, drought signal, the leaf are drooped by ethylene. And the abscisic acid is not like that. Abscisic acid always helps to the plant system to withstand or resist resistant genotype. Resistant genotypes through production of system in the plant for surveying the plant under stress condition. So when applied this abscisic acid or ethylene or any other chemicals, any other
रोहित, इनकी आवाज नहीं आ रही है बिल्कुल।
सर वो
नंदिनी,
डिस्कनेक्ट हो गया सर।
हाँ
कनेक्ट
नंदिनी,
हो गए
आप
सर, वो
कंक्लूडिंग रिमार्क
डिस्कनेक्ट
दो
हो
और
गए हैं
वैलिडिटरी स्पीच दिलवाओ आप।
सर।
नंदिनी,
वो ज्वाइन करने को बोलूं उनको?
ज्वाइन
नहीं
कर
नहीं,
दिया
आप
सर।
कंक्लूडिंग रिमार्क दो।
ओके
उस
सर, ओके ओके।
और वैलिडिटी के लिए
तो
आप उनको बुलाओ, डिप्टी सेक्रेटरी।
ओके 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 सर, मे आई रिक्वेस्ट डॉक्टर प्रगति श्रीवास्तव मैम, Deputy Secretary, Agrimeet Foundation, Bharat, for valedictory remark of Bharat 2025. Over to you, Pragati ma'am. Hello.
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 हाँ पांडियन
Ankit sir.
साहब।
I am in the line. Hello. Can I
ओके
proceed?
ओके, देन कंटिन्यू।
Okay. Plant growth regulators and drought endogenous and content of auxin, tryptophan and cytokinin usually decrease. Okay. The overall ABA is dominant signaling in controlling growth and transpiration, but other factor could also be important. Although recent data show decreased cytokinin concentration in the xylem under drought stress, it is still not clear that all plants first responds in the same way to cytokinin at the concentration found in leaf and gourd cells. Drought stress and upper flowering plants is generally recognized as stress hormones that regulate gene expressions, act as a signal for the initiations of process involved in adaptation to drought and other environmental stresses. Abscisic acid and cytokinin have opposite role in drought stress. Increase abscisic acid, decline in the cytokinin level, favors stomach closures, limit water loss through transpirations under water stress. When the plant will wilt, abscisic acid levels typically rise as a result of increased synthesis. Increased abscisic acid concentrations lead to many changes in development, physiologically and growth. Abscisic acid alter the relative growth rates of various plant parts, such as increase the roots to shoot dry weight ratio, inhibitions of leaf area development and productions of prolific and deeper roots. Ethylene may serve to regulate leaf performance through this lifespan. Proline, action of proline in drought condition, accumulations of proline in plant is all well known to tolerance mechanism from drought stress, which act as cellular asthmatic adjuster and also protect and stabilize essential cell components like protein, photosynthetic apparatus, detoxify reaction oxygen species. Proline is one among the most important cytosolids. Synthesis in leaves at lower potential is caused by the combinations of increased biosynthesis and low oxidations in mitochondria. So proline content were increased under drought stress in pea plant. Proline under drought that acted as an asthma protectant and induced drought tolerance. Then glycine betaine plays an important role in the glycine betaine is secondary metabolites. So primary metabolites, secondary metabolites, under drought condition only secondary metabolites play the major role. This is glycine betaine, so one of the amino acids plays the important role in enhancing plant tolerance and a range of, and was obtained by many workers related to glycine betaine's characteristics. So plant stress tolerance protectant. Already I told asthma regulators what are they. In addition, I am telling some of the things. So proline, sucrose, proline, sucrose, trehalose, and glycine betaine, alanine betaine and proline betaine. Phytohormones are all the hormones, but the stress hormones, abscisic acid, ethylene only worked out well in the drought conditions. Then signaling molecules, salicylic acid, brasinosteroids, zasmonates, H2O2 nitric oxide. So signaling molecule salicylic acid. Salicylic acid always helps to stimulate the secondary metabolites during drought condition. Actually, those genotypes resistant, natural resistance having the salicylic acid giving the value to support to glycine betaines, all the secondary metabolites. With the help of salicylic acid, the plant getting resistance. Then antioxidant. This is the main role. Salicylic acid is giving the value to the secondary metabolites during drought conditions. So particular genotypes getting resistance or tolerance. Then antioxidants, ascorbic acids, luteolin, and alpha-tocopherols, trace elements. So micronutrients and major nutrients are secondary nutrients also helpful to the drought conditions. Then polyamines, they are arginine, ornithine, then spermidine. These are all the plant stress tolerant protectants. These are all the plant stress tolerance protectants. Conclusion: global warming increasing the drought effect on crop productivity. To feed the increasing population stresses, need to manage to ensure food security. So integrations of the molecular approaches, morphophysiological analysis, and conventional breeding strategies will accelerate cultivating the drought-resistant plants for agricultural productions. The future recommendations: the crop plants have involved certain resistance against drought stress. We should identify this mechanism and traits. So need to develop more drought-tolerant varieties. Non-conventional approaches like molecular breeding can play a significant role to build drought tolerance. And genomic regions carrying drought-tolerant genes can contribute for abiotic stress tolerance. So this conclusion, that is my study conclusion. I think no need. So already I told VRMGG1, VMGG, 1205 is the very, and in green gram, 114, 116 in black gram, significantly somewhat better. So with this, I conclude I am very happy. I'm very happy to say, I would like to say thanks to Dr. Ankush Sharma for giving me the opportunity. Even initial, I have some trouble in my mobile. I got my my wife mobile to help during this presentation. I thanks one and all for participate, and also organizers like Ankush Sharma and Nandini ma'am, then Rohit Sharma. They are helping for my trouble during this presentation. Thanks one and all. Thanks for your patience for initial starting presentation. There was some hurdles. So you patiently giving support, giving to me heartful support to all of you.
Thank you so much, sir.
Thanks to participant and organizer. Thanks one and all. Those who are going to
Is
continue
there any option of
plant
remove?
crop species, I think I hope this is helpful to you. Hello.
Thank you so much, sir. We are grateful
Hello?
for your presence.
Thank you, ma'am. Thank you, ma'am.
May I now
Initially, I got
request?
attention.
Yes,
Initially, I
sir.
got
Okay.
tension.
Rohit ji, please do the needful, whatever I am saying you indirectly.
Done, sir. Done.
Okay.
May I now request Dr. Pragati Srivastava, ma'am, Deputy Secretary, AgriMeet Foundation, Bharat, for valedictory remark of Bharat 2025. Pragati ma'am, over to you.
Dr. Pragati ji, are you getting my voice? प्रगति की आवाज नहीं आ रही मुझे.
Should I call her, sir?
हाँ, yes, please, please. 30 दिन में कभी ऐसा नहीं हुआ जो आज ग्लिच ये हो रही है, कभी नहीं हुआ ऐसा.
Yes, sir.
हाँ, लास्ट सेशन में ये होने वाला था कि अंकुर शर्मा की बेइज्जती करानी है, तो वो जूम ने भी कसम खाई है.
सर उनकी साइड से कोई ग्लिच आ रहा है, वो भी ज्वाइन कर रही हैं दूसरे डिवाइस से. Rohit ji,
नंदिनी,
could you please unmute her if she is muted?
1 minute.
आज एग्री मीट का ग्लिच डे है क्या?
I don't know, sir. आज ऐसा ही लग रहा है.
Pragati ma'am, please unmute yourself from the other device from you have joined.
वो अनम्यूट ही है, बट उनकी शायद आवाज नहीं आ रही है, उनका माइक काम नहीं कर रहा, आई थिंक.
उसमें लिखे आ रहा है कि होस्ट इज नॉट अलाउइंग अनम्यूट देमसेल्फ. ये क्या सिस्टम है रोहित, देखो जरा.
मैम बट यहाँ पे अनम्यूट शो हो रही है.
हेलो
प्रगति आवाज आ रही है?
हेलो
हाँ, आ रही है. हाँ प्रगति जी. प्रगति जी,
हेलो
हम आपको सुन पा रहे हैं, आप प्लीज बोलिए प्रगति जी.
ओके सर.
प्रगति जी, आप बोलिए, अब क्या हुआ?
उन्होंने जिस डिवाइस से ज्वाइन किया था, आई थिंक वो लीव कर दिया है.
मैम परमिशन है सबके पास अनम्यूट करने का.
बट उनका शायद माइक नहीं चल रहा है.
सर आवाज आ रही है?
Yes, ma'am. Please continue.
एक मिनट.
Yes, respected Pragati Madam, you are quite audible.
Good evening to all the eminent dignitaries, respected patrons and advisors, distinguished speakers, organizing committee members, and dear participants of the International Winter School Bharat 2025. I, Dr. Pragati Srivastava, is honored to deliver the valedictory address at the close of 30-day journey dedicated to boosting holistic agriculture through research, awareness, and training. Over the past month, Bharat 25 has served as a vibrant platform, bringing together students, faculty, scientists, KVK professionals, policymakers, and practitioners over India and abroad to engage with frontiers areas of agriculture and allied sciences. Through lecture, hybrid session, virtual field demonstration, visual lab work, and interactive group discussions, this winter school has truly embodied its mission of integrating advanced training, interdisciplinary collaboration, and global exposure. The inauguration of Bharat 25 Winter School was graced by distinguished panel of dignitaries whose support and wisdom set the benchmark for excellence throughout this 30-day academic journey. These include our esteemed chief guest, Dr. Dheer Singh, Director cum Vice Chancellor ICR NDRI Karnal, Haryana, and governing body member of ICR New Delhi. Director cum Vice Chancellor, uh, along with guest of honor, Mr. Saurabh Kumar, IFS Secretary, Science and Technology and Renewable Energy, Chandigarh, Dr. Sandeep Kumar Shukla, IIT Hyderabad, Dr. Rajesh Kumar Dubey, Director UGC MMTTC Jodhpur, Dr. Rajiv K. Varshney, Director Murdoch University, Australia, Dr. Anil Dikshit, Joint Director ICR NIBSM Raipur, Chhattisgarh. The program was designed with a clear set of forward-looking objectives to strengthen competencies in precision agriculture, biotechnology, genomics, nanotechnology, AI, robotics, renewable energy, climate smart farming, and digital agriculture, while also deepening understanding of policy framework and food security. It has emphasized interdisciplinary integration across agricultural sciences, food technology, veterinary sciences, agribusiness, and environmental sciences, thereby aligning with NEP 2020 and national priorities for rural agricultural development. Across its thematic areas, Bharat 2025 has highlighted how smart farming, agriculture, digital AI-enabled tools, drones, and IT-based management, farm-based management, and data-driven decision support systems are reshaping modern agriculture. At the same time, modules on soil health, water conservation, pest and disease management, organic and regenerative agriculture, sustainable livestock farming, and renewable energy in farming have reinforced the centrality of sustainability, resilience, and ecological balance. The winter school has also foregrounded agri entrepreneurship, post harvest technology, and value addition, encouraging participants to think beyond toward production, market linkages, startup culture, and rural innovation. Session on policy insights and global perspective have connected participants to government scheme, international best practices, and collaborative opportunities with institutions such as ICAR, ICRISAT, ICARDA, and multiple agricultural universities and in India and abroad. The organizing committee and volunteers drawn from a large network of universities, KVK, and research organizations across the country deserves special recognition for their tireless work in coordination, technical support, communication, and smooth conduct of this hybrid mode winter school. Their effort have ensured that every session from inaugural to technical module to this valedictory was conducted with professionalism, punctuality, and a participant-centric approach. To all the participants, UG and PG student, research scholar, PhD candidates, postdoctoral fellow, faculty, KVK professionals, policy makers, and working professionals, your active engagement has been the heart of Bharat 2025. Your question, reflections, chat interaction, assignments, and article contribution have transformed this winter school from a one-way training program into into a truly collaborative learning ecosystem. I would like to place on record my sincere gratitude to Ms. Nandini and Ms. Sachi for the meticulous and timely execution of every component of this event. Their constant coordination and their quiet efficiency have been instrumental in making this winter school a success. I am equally thankful to Dr. Rohit Thakur and our entire technical team for ensuring the smooth functioning of all sessions from registration and the live streaming of recording and participant interaction without any major interruptions. I would like to offer a special word of appreciation, never but not the least, Dr. Ankur Sharma, Chief Secretary, AgriMeet Foundation and Course Director of Bharat 2025. His visionary leadership, tireless coordination, and unwavering commitment have been the driving force behind this winter school. From conceptualization, the academic framework, to bringing together an exceptional panel of national and international experts. He has ensured that every component of this program reflects both scientific rigor and deep concern for the farming community. His constant encouragement, calm problem solving, and personal involvement at every stage have truly inspired all of us, have set a very high benchmark for future academic initiatives. Thank you, Dr. Sharma sir. In order to meaningfully assess the learning outcome for this winter school, a 2-day online test will be conducted. This is an important announcement. Will be conducted on 29th and 30th of this month. The test link will be shared in the official participant group, and all the registered participants are encouraged to appear for this assessment. Performance in this online test will be considered for the final evaluation, and certificates will be issued accordingly based on successful completion of the program and the assessment. Before concluding, I would like to make an important announcement. It's a pleasure to announce two important upcoming events. The first is a 10-day international workshop cum short-term course with a nominal fee structure will be organized from 5 to 14 December 2025 on the occasion of World Soil Day, focusing on modern science-led school and agricultural innovation. The second is 7th Global International Conference that will be held in the Union Territory of Chandigarh at the Chandigarh Secretariat in offline mode in the upcoming year on 2021 from 20 to 21 February 2026, providing yet another prestigious platform for scientific exchange, policy dialogue, and networking in agriculture and allied sciences. You all are welcome, warmly welcome, invited to participate and share all these opportunities within your professional networks so that learning momentum of Bharat 2025 continues to grow in the months ahead. Thank you very much for your cooperation. Thank you very much. Over to you, Ankur sir.
थैंक यू डॉक्टर श्रीवास्तव। इतनी टेक्निकल ग्लिच के बाद भी आपने बहुत अच्छा वैलिडेटरी रिमार्क दिया। सभी पार्टिसिपेंट से मैं पर्सनली कि मैं कह रहा हूं कि मैं गुनहगार हूं, मैंने गलती करी है कि जो भी टेक्निकल ग्लिच आज हुई है मैं अपने ऊपर ले रहा हूं। आप मुझे भर भर के सुना सकते हैं, मुझे मेल्स लिख सकते हैं, व्हाट्सअप कर सकते हैं, इस गलती की पूरी जिम्मेदारी मैं लेता हूं। लेकिन मैं इतना भी कहना चाहूंगा कि इतने बड़े बड़े प्रोग्राम्स जब होते हैं तो इस तरीके की टेक्निकल ग्लिचेस हो जाती हैं क्योंकि हम लोग ऑनलाइन पेडागॉगी में हैं। लेकिन बहुत जल्दी हमने आपके लिए एक ऑफलाइन इवेंट्स को भी निकाला है, ऑफलाइन इवेंट को भी डिजाइन किया है, जो 20 से 21 फरवरी में गवर्नर सेक्रेटरीट, आवाज आ रही है मेरी?
जी सर।
चंडीगढ़ सेक्रेटरीट में 20 से 21 फरवरी में हमने एक डिजाइन किया है प्रोग्राम, जो हमारी 7th इंटरनेशनल ग्लोबल कॉन्फ्रेंस है एग्रीमीट 2026। तो जो हमारे पार्टिसिपेंट्स हैं आज जो भारत 2025 का हिस्सा है, मैं सभी से रिक्वेस्ट करूंगा कि आप चंडीगढ़ आए। चंडीगढ़ की ब्यूटी आप सब लोग बहुत अच्छी तरीके से जानते हैं। पास में आपका हिमाचल है, पास में आपका जम्मू एंड कश्मीर है, तो आप चंडीगढ़ घूमिए, इससे जुड़े हुए स्थानों को घूमिए। एक अनाउंसमेंट जो हमारी डेप्यूटी सेक्रेटरी ने किया है कि 2 दिन का एक यूजीसी ने ही ऐसा निकाला है कि आप अगर कोई विंटर स्कूल या कोई कोर्स या कोई वर्कशॉप या कोई शॉर्ट टर्म कोर्स कर रहे हैं तो एक एग्जाम जरूर होना चाहिए। तो आप प्लीज ये भ्रमित मत होना, ये कंफ्यूजन मत लाना कि ये एग्जाम यूपीएससी लेवल का है। 10 क्वेश्चंस होंगे सिंपली, बहुत छोटे, जो आप बहुत आराम से कर पाओगे। 10 क्वेश्चंस को करने के लिए 10 मिनट आपको दिए जाएंगे। गूगल फॉर्म पे क्वेश्चंस होंगे, 1 2 3 4 ऑप्शंस होंगे, एक ऑप्शन आपको टिक करना होगा, ठीक है? तो 10 मिनट का एग्जाम है, ये सैटरडे को भी होगा और ये संडे को भी होगा 10-10 मिनट का। तो लिंक पे आप आएंगे, ठीक है? और 6:30 से 6:40 होगा 10 मिनट के लिए। 6:30 to 6:40 सैटरडे को भी और 6:30 to 6:40 आपका संडे को भी। तो इससे 30 डेज पूरे काउंट भी हो जाएंगे। सॉरी, मुझे बहुत सीवियर कोल्ड है, इसलिए मुझे ज्यादा बोलने पर खांसी आ रही है, इसको इग्नोर कर दीजिएगा। तो ये क्या है कि 30 डेज हमारे पूरे कंप्लीट हो जाएंगे। एक तो यूजीसी की प्रोटोकॉल भी पूरी हो जाएगी क्योंकि यूजीसी इज वन ऑफ द मेजर कोलैबोरेटर इन दिस 30 डेज विंटर स्कूल इनिशिएटिव। ठीक है, एंड सिग्नेचर भी डायरेक्टर यूजीसी के इस पे होंगे। तो ये तो एक आपसे जुड़ा हुआ विषय हो गया। मैं पुनः टेक्निकल ग्लिच के लिए आप सभी से हाथ जोड़ के माफी मांगता हूं कि ये जो चीज हुई है ये नहीं होनी चाहिए थी, और आगे से भी हम ध्यान रखेंगे कि चीजों को थोड़ा सा और डिजिटल टेक्नोलॉजी, हमारे प्रधानमंत्री कहते हैं डिजिटल भारत, इसके बावजूद भी हम डिजिटलाइजेशन पे इतने पीछे हो रहे हैं, ये मेरी कमी है, फिर से सॉरी। तो हम सभी कैंडिडेट से आग्रह करते हैं कि कल 10 मिनट के एग्जाम को आप लोग जरूर अटेम्प्ट करें, और बहुत जल्दी हम लोग माटी 2025 जो हमारा वर्ल्ड सॉइल डे पे इनिशिएटिव है, 10 डेज का सर्टिफिकेट कोर्स है। बहुत से कैंडिडेट्स, बहुत से स्टेट एग्रीकल्चर यूनिवर्सिटीज, सेंट्रल एग्रीकल्चर यूनिवर्सिटीज, इवन एएसआरबी सिस्टम भी 10 दिन के सर्टिफिकेट कोर्सेज को कैरियर एडवांसमेंट स्कीम, कैश प्रमोशन और डायरेक्ट रिक्रूटमेंट के लिए मांगने लगे हैं। तो उन कैंडिडेट्स के कारण इस कोर्स को डिजाइन किया गया है। आप उसमें रजिस्टर कर सकते हैं। मिनिमम फीस 150 है, लास्ट फीस उसकी 500 है। नेक्स्ट इवेंट हमने बताया है जो हम चंडीगढ़ सेक्रेटरीट में 20 एंड 21 फेब को कर रहे हैं। आप उसमें अपना पार्टिसिपेशन करें, ओरल और पोस्टर प्रेजेंटेशंस दें, एकेडमिक अवार्ड्स के लिए अप्लाई करें, स्टॉल लगाएं, ठीक है, एग्जीबिशंस लगाएं, सबको हम ओपन हार्टली इसमें इनवाइट कर रहे हैं। एंड अगेन आज की टेक्निकल ग्लिच के लिए बहुत बहुत क्षमा, और सभी पार्टिसिपेंट्स को सक्सेसफुल विंटर स्कूल के एग्जीक्यूशन की बहुत बहुत शुभकामनाएं। मैं डॉक्टर अंकुर शर्मा इस विंटर स्कूल भारत 2025 के समाप्ति की घोषणा करता हूं। सभी को बहुत बहुत धन्यवाद। जय श्री राम, नमस्कार, गुड नाइट एवरीबॉडी। All participants can leave, please.